Fighting Blindness Canada's Viewpoint Education Series shares the latest in vision research, provides opportunities for you to ask your questions, and connect with other people who are living with a blinding eye disease. The session you're about to watch is a recording. To learn more about the research we fund and upcoming programs and events, please visit our website at fightingblindness.ca. Viewpoint is proudly presented by our Platinum partner Bayer and supported by our Gold partner Roche and Silver partners AbbVie, Astellas, Beacon Therapeutics, Johnson & Johnson, and Specsavers. Thank you for watching. We hope you enjoyed this session and share your thoughts with us in the comment section below.
All right, everybody, we're going to get started. Good morning, good morning. My name is Morgan Einesen, and I am the Senior Manager of Education at Fighting Blindness Canada. It is my absolute pleasure to welcome you this morning to our Viewpoint Toronto Speaker Symposium. Thank you so much for joining us. We're so glad that you're all here for what promises to be a wonderful day of learning and connection. And this is a full room. It's very exciting. So for those of you who may be new to us and our community, Fighting Blindness Canada is the largest charitable funder of vision research in the country. And now for over 50 years, we've been working to help advance research, provide education, and offer support to people and families who are affected by vision loss. I want to start us off today by acknowledging the Indigenous people of all the lands that we are on today. Here at North York Central Library, we are on the traditional territory of the Haudenosaunee Confederacy, the Wendat, and the Mississaugas of the Credit First Nation. This land is also part of the Dish with One Spoon territory, a treaty between the Haudenosaunee Confederacy, the Anishinaabeg, and allied nations, to peaceably share and care for this land, its waters, and all of the biodiversity in the Great Lakes region. And because we have people tuning in from across the country, we acknowledge all of the Indigenous people that call this land home. So before we move into the program, a couple of quick housekeeping notes. The washrooms are located on the side of the room. So if you're facing the stage, they're on the far left side. Just to be aware, there is a staircase also kind of in the middle of the room on that side. So just be aware of that. And we do have volunteers sort of standing by to make sure you can find where you're going. At the very back of the room where you came in, hopefully you've already had a chance to take a look. We have light refreshments, coffee, and some pastries. And then in your event bag that you got when you came in, you should have a copy of a physical printed program. There's also a QR code in the middle of your table if you'd prefer to access that on your phone. Our amazing volunteers who are here are stationed about the room today. So if you do need any assistance, just give us a wave and we'll come to you. We have such an exciting morning ahead of us. I wanted to say that we have two sort of sessions. The first session is going to be our keynote with Dr. David Gamm, the star of today's show. And then after that, we'll be having a panel discussion with Dr. Abu Baker, Dr. Kunakoop, Dr. Gamm, and a community member, Bernie Zink, all about clinical trials. So to officially kick things off, I want to invite our President and CEO, Jennifer Jones, to join me on the stage and to share a few opening remarks.
All right. Hello, everybody. We being my team, had an event already this morning with our corporate sponsors, many of whom have joined us for this event. Dr. Gamm spoke to us, and you are in for a treat. I was teasing him that stem cell research tends to be very sexy in this sector, and I think the turnout today is an example of that. So I thanked him for bringing sexy back to the vision loss sector. It's a thrill to see so many of you joining us today, especially on a Saturday morning. In a landscape where there are many organizations working to support vision health, we are deeply grateful that you've chosen to give us your time, especially on an early morning. We believe that eye disease research is where the most meaningful progress happens. At Fighting Blindness Canada, we take pride in focusing on what we do best, driving progress in eye disease research and improving the lives of people living with vision loss. Based on the turnout today, I'm going to assume you probably feel very much the same. Before I jump in with a little bit more background, I'm really curious how many people here have joined us for the first time. Could you raise your hands? Like really, wow. All right. That's a really good chunk of the room. Welcome to Fighting Blindness Canada. Welcome to this community. It's an incredible group of people. I hope you feel that your time is well served by being here. One of the things I also like to start with, and this is a little bit more of the human-centric side of things, is get an idea of what people want to get out of this day. This is designed specifically for you. We have the expertise, but we want to make sure that we're bringing it to you in a way that is meaningful. Would anyone want to share one reason why they're here? Is it because you have a question about an eye disease? Is it because there's someone that you want to meet, not just Dr. Gamm? Does anyone want to share why you're in the room and what you hope to get out of the day?
This is the whole
Nita Reed.
Need to tighter on the yeah. I think that's how it looks on the webinar.
Yeah.
Tighten it on the podium.
Thank you. We're thrilled that you're here with us. Anyone else want to share what they want to hope to get out of the day? No? Okay. Then I'm going to move on and say again, though, welcome to everybody.
This is
I
what
also want to take a moment to say thank you to our sponsors. Without their support, events like this would not be possible. Right? I want to start with our big thank you, our platinum presenting sponsor, Bayer. Also, our gold partner, Roche, and our silver partners, AbbVie, Astellas, Beacon Therapeutics, Johnson & Johnson, and Specsavers. Lastly, yes, thank you. Thank you, Nita. You are right to give the applause to them. They are an incredibly essential part of our ability to do this work and bring it to our community across Canada. I also want to take a moment to say thank you to our volunteers. We have quite a few in the room here, so you should always feel that you can lean to them if you need help getting food, going to the washroom, or need to navigate the room. Thankfully, everyone who had signed up is here, because if not, you would have been assigned a role, because we really do need volunteers to make this event run smoothly. Also, lastly, FBC is 100% funded by philanthropy. We don't receive any government funding, and so we are very much in deep gratitude to all our donors and our supporters. If you're interested in getting involved as a volunteer, as a supporter, or as a donor, there are many ways that you can make a difference in the work that we do. And before I introduce Dr. Larissa Moniz, who's going to speak with Dr. Gamm, I also want to say that we hope that you leave today feeling that your time has been a really smart investment, an investment in your health, an investment in the information that you are receiving, and an investment in the connections that this room offers you. Again, thank you for joining us today, and I want to introduce Dr. Larissa Moniz.
Hi, everyone. I'm Larissa Moniz. I'm the Director of Research and Mission Programs at Fighting Blindness Canada. I'm just going to take 1 minute so I could introduce our keynote speaker, who really is why we're all here today, to hear him and our wonderful guests. So Dr. David Gamm, he is the Director of the McPherson Eye Research Institute, the Trout Chair in Eye Research, and a Professor of Ophthalmology and Visual Sciences at the University of Wisconsin-Madison. In 2016, he founded Opsis Therapeutics, where he serves as Chief Scientific Officer. In his practice, Dr. Gamm diagnoses and treats pediatric eye disorders, while his research focuses on the use of human pluripotent stem cell technology for retinal disease modeling and cell replacement therapies. And Fighting Blindness Canada has very proudly supported Dr. Gamm's research through a Restore Vision 2020 grant over the past few years. And we were really, really thrilled to see this research develop into a photoreceptor cell therapy that has now moved into clinical trials this year. So I want to thank Dr. Gamm for coming all the way from the States, very late flight last night. He's out pretty early this afternoon, so doing a lot of work for us, and we really appreciate his effort. And I'm looking forward to hearing all about his research.
All right, thank you, everybody. It's great to be here. Foundation, or I could say FFB Canada, which is what it was used to be called. That tells you how far back I go with the organization. But Fighting Blindness Canada was one of the original foundations that supported the work that we did. It was critical in developing the portfolio that we needed to go to industry to be able to fund ultimately the manufacturing and clinical trial that we're involved in right now. So I'm deeply indebted to the folks of Canada, the people that support FBC. And while I'm only here for a day, the one-day respite from the insanity down south is worth the trip by itself. So I do appreciate that. But, okay, so and I'm going to do my best to describe what I'm talking about. And I'm not always the best at doing that, but I will try. So kind of how I envision myself in a nutshell, I'm from Wisconsin, we live in Wisconsin, so I think of myself as a retina farmer. So our goal is to be able to grow or to develop cell types that can be used for two purposes. One would be to replace those cells that are lost during the course of disease, in this case, retinitis pigmentosa, other inherited retinal degenerative diseases, or to use the cells that we make to test gene therapies, genome therapies, and be able to advance them, all with the ultimate purpose of being able to help patients who have otherwise untreatable conditions. Okay, so let's see what we do that. So a couple disclosures. I just want to be clear with where my conflicts are so that you can ignore the things. You say, oh, he's got a conflict there. He's probably just going to make that stuff up. But so I do have patents on retinal organoid technology. So the ability to make retinal tissues and cells from human pluripotent stem cells is something we patented way back in the early 2000s. And I am, as a basis of that, as was mentioned, I have an involvement with Therapeutics, which is involved with Fujifilm, and we have partnered with Blue Rock Therapeutics, which is a subsidiary of Bayer, and I'm a consultant for Blue Rock Therapeutics as well. Okay, and so what does my lab do? I kind of mentioned this already, but we use human pluripotent stem cells, and I'll talk a little bit about what that is in a moment. And we do a number of different things with that. First and foremost, we're kind of developmental biologists. We want to know how a photoreceptor is made. I'm essentially a cell manufacturer. So I'm interested in how a cell that can become anything that it wants to be, can grow up to be anything it wants to be, how it decides to become something as complex and specific as a photoreceptor cell, and how does it work. And so we have unprecedented abilities to be able to do that in a dish where we can watch every step along the way using pluripotent stem cell technology. And if we're successful in doing that, and we can genuinely produce authentic human retinal cell types, which is otherwise impossible to do, then we have the capacity to use them in applications to help patients. So either cell replacement or in developing genes and other types of therapies. Okay, so what I'm going to do today is I'm going to arrange my talk in the form of a fact sheet. So frequently asked questions. So, and working with patients and talking to folks, I get a lot of the same questions about stem cell technology and whether it might be right for them. And so the questions that I'm going to go through today, one by one, are first, is retinal cell replacement right for me or my loved one? What retinal cells or cells do I need? I mean, there's no silver bullet here. There's not like one size fits all. Where do these cells come from? Where do they go? How do they get there? How do clinical trials work? How many clinical trials can I participate in? And are there risks? Oh, and then lastly, why does it take so darn long? So, those are all the questions that I'm going to go through one by one. All right, to start out with, we're going to say, is retinal cell replacement? Probably the biggest one, right? Because there's a lot of things out there, thankfully, for patients. I mean, 20 years ago when I first started doing this, there was nothing really. So, but now, careful what you wish for. Now there's so many things that you get inundated with, what's right for me or my loved one. And so it really depends. I think the easiest way to look at it is how much retina structure and function you have left. So it's not magic. Tissues are made up of cells. They're building blocks. They're connected to one another. It's like a layer cake. And so if you're replacing a cell type or replacing one part of that cake, you kind of need the rest of the cake there. Otherwise, you're putting a brand new engine into an old rusted car that doesn't have wheels. It's maybe a beautiful engine, but the car is still not going to make it to the Tim Hortons. So, so you have to kind of think where you're at. So in a disease process, early stage folks that maybe just got their diagnosis, they're doing well. Their vision's good. They can get around, do all the things that they want to do. A little later on, you start to sense something's up. Maybe you're not seeing as well in the evenings. Maybe you're starting to get a little blurry, or you're having some visual field issues. And so you're aware of things. You're starting to see deficits arise. And then you go to late stage disease where you have profound vision issues. And so early on in that process, really, the focus would be on gene therapies, because what a gene does is you're replacing one gene in an otherwise possibly functioning cell. So you're trying to replace the furnace in a house that's cold, but the house is still standing. Towards the end of that disease spectrum, when the cells are no longer there, if you arrive at the doorstep with a new furnace, the house is gone. So in that case, you really have to look for other cell types or other therapies like cell therapies. And there's other optogenetics, things like that can be considered as well. So knowing where you are on that spectrum of disease is really critical to know what general categories of therapeutics might be right for you. Just to show you that it's just not cartoons. These are actual pictures. It's called histology of a human retina that I'm showing right now. And so what I'm showing is different layers of individual, like circles, but they're individual cells or parts of cells. And it's the layer cake of the retina. So if we take that out of a human being and we blow it up, look under a microscope and do special stains, we can see all the different cell types that are connected and form the circuit within the retina. And so early on this, I'm showing on the left, a nice thick photoreceptor layer, the purple plus the pink at the bottom. And then a little bit later on, you see it's starting to deteriorate. Now you physically see that it's getting thinner. These photoreceptors are starting to die off. But there's still enough there where this patient probably still sees pretty well, probably doing everything they normally would do. So you can take a hit. There's redundancy built into the retina such that you can lose a lot of cells and still not even know what's going on. But then later on, now they're almost all gone. And then later on, it starts, the cells, the rest of the layer cake starts to deteriorate. So it's not something that we're just talking about in ethereal terms. It's biology. We can actually see this in a live patient using technology that's available in nearly every ophthalmologist's office. So again, early on, gene therapy would be the focus. Later on, you're looking more at cell therapies because you have to replace the cells. There's no longer a cell there for a gene to be expressed in or a drug to operate on. So that's what I'm going to talk about is the cell therapy end of things. So that's really the main thing to know about whether cell therapy is right for you, is to understand and to talk to your providers about where am I on that spectrum. Okay, the next question is then what retinal cell or cells do I need? And for this, I'm going to go back to, this time, a cartoon that shows the different levels. So before I showed an actual picture of a human retina, but this is a cartoon of that, which shows those layers. And deepest within the retina are the light-sensing cells, or the photoreceptors, and they're up against their support layer called the retinal pigment epithelium. And they're kind of the handmaidens. If you think of the photoreceptors as the divas of the retina, they get all the credit, but they can't even brush their own teeth. They need other cells to help them out. And the main one for that is the retinal pigment epithelium. And then they connect to a layer higher to them called bipolar cells, and they connect to a layer higher to them, which has ganglion cells, and those ganglion cells send their projections through the optic nerve back to the brain. Okay, so it's a circuit. It's like a three-wire circuit. And so in primary photoreceptor degeneration, there's a genetic condition that causes those photoreceptors to first become dysfunctional, not work very well, but ultimately, if it's not used, it starts to deteriorate over time. And so you frankly don't have those photoreceptors anymore. They're the tip of the spear of your visual system. If you don't have photoreceptors, light comes in, there's nothing for them to hit. Essentially, you don't see anything. You presume, however, that the rest of the circuit downstream of that is relatively intact. Okay, so in that case, what do you need? You would need photoreceptors, right? That's probably the most simple case. And this is a very stylistic situation that I'm providing for you. So another very common type of retinitis pigmentosa, or inherited outer retinal degenerative disease, doesn't affect the photoreceptors primarily, but affects that helper layer, that retinal pigment epithelial layer. And so when that happens, you need RPE cells for sure, because the RPE cells go away. But unfortunately, what happens pretty quickly after the retinal pigment epithelium goes away is that the cells that they subserve, the photoreceptors, they also deteriorate. So if you get into a type of blindness where it's primarily the retinal pigment epithelium early enough, then you might need just an RPE cell. But typically, if it's later on in the disease, you need two cells. You need the RPE cells, and then you need the photoreceptors that died secondarily to the RPE being lost. Okay? But that's not the end of it. So as I kind of mentioned before, over time, if those parts aren't working, the rest of the retina that is not really doing anything starts to remodel. Maybe it gets scarring. It sits there kind of inactive for a long time. And before you know it, you might very well need an entire retina. Okay, and so what we're talking about now is replacing one cell or two cell types, and that's what we're doing. But we're not talking about replacing a whole retina. I also am part of a big consortium that's looking at whole eye transplants. That's a whole nother issue, right? But so different people need different things, different spare parts to be replaced. Okay, third, so where do the cells come from? So we understand the process. We understand now more and more the genetics that lead to the cells dying. We understand more and more why the cells die, and that's very important. But if you don't have a source of replacement spare parts, you're kind of out of luck anyway. And so that's where you kind of start back in way before I started in the field. People looked into, well, living or deceased donors. What happens if your kidney deteriorates? Well, you go get another kidney, you get a donor. So people did look at whether or not you could take another living donor or a deceased donor, take a photoreceptor out of them and see if that would potentially plug back in and work. Problem is none of that really did work, and the reason for that is that if you take a mature photoreceptor, they're very fragile. They already have their connections made, and so they don't reintegrate. They tend to die, and even if they survive, they just sit there and they really don't plug themselves back into the system. So living or deceased donor, and also obviously it's hard to get those. So, and if it's a living donor, you kind of have to blind yourself in one eye to provide those cells to your loved one or somebody else. And so that's a tall ask. What about a non-human donor? I mean, we do pig valves sometimes, and sometimes we're able to use other non-human species for it. And similarly, it doesn't work. So what about lab manufacture? What if we can grow them? And that's where really the holy grail was, but there was no ability to do that until, this is my plug for the state of Wisconsin, in the late 1990s when Jamie Thompson, I'm very fortunate to be good friends of him he's recently retired, lives in Washington State now, but he was the one who developed human embryonic stem cells. And so he was on the cover, I'm showing a picture of the cover of Time, where he hated this picture, he didn't want to be part of it. He's a very, he's an introvert, but anyway, I always show it for regardless, and he hates it. But he developed the ability to be able to produce human embryonic stem cells, and that, for the first time, provided the clay or the source that at least theoretically had the potential to make any cell type in the body. And so that's where I want to take a little bit of a segue in terms of using the word stem cells. And so it's thrown around a lot, oftentimes for ill-gotten purposes, because people think there's something magic about them. And so not all stem cells are the same. They're actually not brand new. They weren't invented in the late 1990s by Jamie Thompson. He just figured out a way to grow them from human sources. Stem cells, we've known stem cells have been around forever. We all originate as a fertilized ovum, and so all of our cell types come from a common clay, a common cell type. So that's been known in developmental biology for eons and eons. But it's a matter of there are different types of them. We have bone marrow stem cells. I'll give you some examples here. And they're in our bone marrow because our blood cells only survive, our red blood cells survive about 3 months and then they die. So if we don't have an ability to make more blood cells, we have aplastic anemia and we die. So there's also stem cells in your skin that create new skin. There's stem cells in your fat. And people will just kind of use that term. They'll say, well, I'm going to give you some stem cells. And in a famous case, it was actually fat. And they stuck it in somebody's eye because they convinced them that it was a stem cell. And guess what it did? It made fat in their eye. So again, it's not magic. It's biology. It's rooted in good science. And so you have to be able to understand that so that you don't get fooled. And there's also stem cells or progenitor cells that can be taken from fetal tissue as well, too, with, of course, the ethical issues that go along with that. But none of those actually were effective in being able to authentically replace photoreceptors in a human retina. So it really was pluripotent stem cells. So it's that term pluripotent is important. So pluripotent stem cells are the only cell types that are capable of producing the cells that we have in our own body. Now, it doesn't mean that they can do it equally well, that we have the ability to make every single cell type, but that's what we started with. And there's two types, the embryonic stem cells that I mentioned before that were originally discovered by Dr. Thompson in the late 1990s, but then in the mid-2000s, both Dr. Thompson at Wisconsin and Dr. Yamanaka in Japan figured out a way to take skin cells or blood cells or other cells that we have as adults and genetically reprogram them back to an embryonic stem cell state. And so that's what we use now. So we use cells that are taken from adults, and we use those to do all of the things that I'm going to talk about from here on out. Both of them work well. It just happens to be that our choice has been to use iPS cells. And this is what they look like. So I'm showing a very nondescript colony of pluripotent stem cells. They're not much to look at. They're round cells. They look gray on this particular image. And there's thousands of them there. It's not just, that's not 2 cells. That's actually 2 colonies with thousands of cells in each one of them. And so the hard part then is to be able to harness the power of a stem cell, which can produce any cell type in the body and will, all glom together in a dish. And so you've got beating heart cells here and some cartilage here and all this sort of stuff. But what are you going to do with that? So you have to be able to direct them towards the cell type that you're interested in my case, retina, be able to extract them, study them. And so that was what took a lot of time in the early 2000s in my lab to be able to figure that out, because we just want the retina. We don't want the rest of the stuff. God bless them for all the other stuff they want to make, and it's important for diabetes, for heart disease, but that's not what I do. I'm an ophthalmologist. And so we worked over the years to be able to develop a system whereby we could extract in 3 dimensions. So we would make these retina-like aggregates. We called them retinal organoids. They make full retinal tissues, and then you had to grow them. They actually grow and differentiate and mature just like they would if they were in utero. So it's not, again, it's not magic. It's rooted in developmental biology. You start with something that looks like a primitive retina, and then it grows and develops and makes all the cell types and the connections that would normally be made if a child was in utero and before they were born. So you end up with a process where you have these small retinal organoids that have all of the parts that we have in our own retinas. Forward. Okay, so on the left I'm showing a lot of these organoids that have these retinal structures, but it's one thing to say that they're human retina, but you have to show it. You have to show it by virtue of what do they look like and how do they function. And so we then embarked on a number of years of study to be able to understand what's inside of those cells, what's their structure look like, and what can they actually do. Okay, and so on the right, I'm showing a picture of cone cells that we grow in the lab. So this was started as embryo or induced pluripotent stem cells, iPS cells, and they've never seen anything outside of a plastic dish. So they're totally grown within the laboratory. If we compare that to actual cones, the cone photoreceptor cells that are in your eyes and my eyes, that's I'm showing you on the left. Now, no one's going to confuse the two. One of them is a lot prettier than the other, right? So my analogy here is I'm a human. George Clooney is a human, right? We both do human things, but I'm like on the right, and George Clooney's like on the left, right? But if you're blind, I'll do, right? So that's but it's more than just saying what the structure looks like. You really also want to be able to say, can it do the job? So there are certain parts of photoreceptors that have specific jobs that are done nowhere else in the body. They have outer segments, which are the antenna that detect photons of light and initiate the whole cascade of vision. And so we were able to show with collaborators at the UW-Madison that they could detect light at a level that is nearly identical to what the cones in our own eyes by using some monkey work that we did by looking at the cones in adult monkeys. We could show that they were nearly identical to what we could grow in a dish. And that's detecting different colors of light, how fast they do it, how repetitive it is, a lot of different parameters that we had to look at. Next, pound for pound photoreceptors are the most energy consuming cells in your entire body. So we need to make sure they can take a punch, that they have the ability to make energy quickly and consistently over time. We showed that with another group at UW-Madison. And then if we're going to take these cells out of these little organoids and put them into a patient, they're going to have to regrow their wires. They're going to have to reach out and connect to the cells that are existing in your retina. And then they have to make connections too. And so we went through a series of studies over the course of about 5 to 6 years, much of which was funded through the Fighting Blindness Canada, to be able to convince myself and to convince our academic and corporate partners that it had this ability and it could do it on its own. So we're not priming them in any way. These are intrinsic autonomous characteristics that these cells just can do on their own. We're not coercing them to do it, they just have that capacity. Okay, so that's all in the laboratory. So we could do that in small batches, and we're doing it by hand and we're, culling out the ones that don't look right and do look right. That's not commercial manufacture. That's not good enough to get into a patient. You have to be able to do it at a much larger scale. You have to be able to do it in a reproducible fashion. You have to pass all this muster for good reason, because if we're putting it into a patient, if we're putting it into one of my loved ones, I want to make darn sure that what I think I'm putting in, I'm actually putting in. So that's a process that's very hard to do in a laboratory, because it takes a lot of funds and it takes a lot of expertise coming from many different directions. And so that's the bottleneck in the process of commercial manufacturing between going from a laboratory to going to a clinical trial. And so number one is you want to be able to show that in the process of converting that to a commercial scale that you have not lost authenticity. You haven't lost the goal at the end of the rainbow, which is to make an actual cell that can do the job potentially in a human patient. But then there's all these different other parameters that you have to hit to make it a viable therapeutic product. And so that's when we started Opsis Therapeutics, because I knew that I needed to be able to take what I did in the lab and be able to partner, in this case, with Cellular Dynamics International, which was a company that had been started by Jamie Thompson in Madison and was subsequently purchased by Fujifilm. So they had a tremendous amount of manufacturing expertise that we could meld with our kind of artisan expertise for making specifically photoreceptors. And then we were able to use that to then eventually scale this up. This is a 3-liter bioreactor, and we can do those in tandem. And what you're seeing are those cells, and there's billions of them. There's enough to treat. I don't know what the equivalent in Canada of Corpus Christi, Texas is, and I don't know why I keep saying Corpus Christi, but it's about that many patients that you can treat. And then we can do this, we can cryopreserve them, we can put them in vials, and do it in a manner that's FDA compatible. So that took years. That took about 3 to 4 years to be able to convert what we did in the lab to something that actually was a lab manufactured, authentic photoreceptor cell. And that really set the stage for us to be able to partner with Blue Rock Therapeutics, which is a subsidiary of Bayer. Okay, that was the longest one, which is where do they come from? Where do they go? So, you might think about, okay, do I rub it on my forehead? Do I, inject it into a vein? Do I inject it in the middle of my eye like you might treat wet macular degeneration? The answer is no. I mean, again, it's not magic. So these are cells that have to go to the lowest layer of that layer cake, which means you have to do a subretinal injection. You have to make a small hole in the retina, you have to lift it up and put the cells where they need to go. They're not going to work their way through there. They can't plinko down through a dense tissue and find their way to go. So again, there are some places that or therapeutics that do that, but they have no way of replacing the photoreceptor. I mean, again, it's just a matter of knowing the biology. So subretinal is the way to go. Right now, what I'm showing here is the two approaches that everyone wants to do. It's called intravitreal, which is injection into the center cavity of the eye. That can be done at bedside. That can be done in the clinic. It's really easy, and the patients love it, and the clinicians love it because anybody can do it. But it's never going to help. It's never going to replace the cell. It can't. So you have to do the harder thing. People don't want to. It's tough. You just have to do it. So the subretinal is the way to go. All right, and then so how do they get there? So how do we get them to that subretinal space? That's where the eye has a definite advantage, and that is that most of the techniques, the surgical techniques that are necessary to do subretinal injections are, I wouldn't say mainstream, but they're pretty common. And there's plenty of retina surgeons with gene therapy expertise that know how to get things to the subretinal space. And so that's de-risked for doing these types of therapies. Now, making a hole in the retina and then placing things underneath it does give you a risk that there might be a retinal detachment, so we have to watch that very carefully. But as the years go by and we have more experience with gene therapy, we understand what those dynamics are like, and it's improved safety of doing subretinal injections. But it's a point of risk for doing a therapy like this. But not only is the surgery relatively mainstream, but all of the postoperative follow-up and the monitoring. Most of the techniques that we use are commonplace in an ophthalmologist's office. And for that reason, no one has to invent a new machine to look at where the cells go. No one has to invent a new instrument to be able to do it. And that also lowers the price of getting to clinical trial, and one of the reasons why so many of these new therapies start in the eye. Okay? I do want to mention, because part of what FBC has been wonderful about supporting are the use of scaffolds. So how do they get there? Subretinal, do we use just cells, or do we put the cells in something to give them an order? And so we've worked on both of those. And so we've work with engineers at UW-Madison to develop biodegradable scaffolds that put those photoreceptors in little compartments that allow us to place the cells exactly where we want them to be and in a better orientation. But there's a price to pay with that, because now you're adding more complexity, you're adding a device, the cost goes up. You can't cryopreserve something that's this complex. So you're always robbing Peter to pay Paul. So ultimately, this is something that can be done in the future, which may very well be a version 2.0, 3.0 that will improve outcomes, but it's not a place to start. And that's what we're doing now in the lab, is we're looking ahead and saying, well, what's the next frontier for delivery? But it's pretty cool, and that's what we're showing you here, especially for patients who need both the RPE cells and the photoreceptor cells. Delivering both of them at the same time can be difficult, especially and keep their correct orientation. So I'm showing here one of those wells. Now if I go back, does it work or no? Yeah. Well, I'm showing one of those wells. So this is a hugely magnified image, and all of the green outlines are individual retinal pigment epithelial cells. So they're the support cells. And then what we can do is we can place the photoreceptors inside of those. So now you have a way to deliver both of those cell types in close proximity to one another. But again, the complexity goes up, the cost, all that sort of stuff has to be considered as well. And what I'm showing here is the injection. It's a little bit no one's eating, so I think we're okay. This is in a pig, and this shows injection of that scaffold with the photoreceptors and the RP into the subretinal space of a pig. And you can see, or I shouldn't say it, but what I'm showing is that there is a kind of an area where we've made an incision into the retina to be able to access that space. And so that afterwards it's lasered so that it can be sealed up. Okay, that's how they get there. And then how do clinical trials work? I'm going to kind of go through that. You might hear about different phases, a phase 1, a phase 2, a phase 3. So first of all, you have to get approval for testing. So you have to do all the preclinical work to pass muster so that the local regulatory agencies or the national regulatory agencies will give you the green light to go forward. That's mostly safety, but you also have to show that you have a chance of working. You then go into what's called a phase 1 trial, and that's for safety only. And that can be between 5 and 50 participants. Typically these days, we don't see pure phase 1 trials. It's usually a little bit of efficacy they're looking at as well. But these are small, and they're essentially done in patients that have the least to lose, so they have the most vision loss. And it's just established to make sure that we don't do harm. And then you move into a phase 2, and it's still safety. Safety is important in every phase of a clinical trial. But now we're looking at dosing. How much do you need? So now it's kind of ramping up. How many cells do you need? Do we put in more? What's our limit in terms of how many cells we can put in to get a positive readout? And then a phase 3 study is the real big one. So it can be 50 to 100 patients in phase 2. Phase 3 can have 100 to 1,000 participants, depending upon how many patients are out there. And that's really looking for, again, safety, but also efficacy. Because now you have the numbers where you can start doing statistics. Earlier on with smaller numbers, you can get anecdotal changes, but some are going to help, the therapy is going to help some, but not others. It's always going to be the case, because we can't fix the type of patient we're putting it into. Everyone's different. Everybody who has a particular condition is different than the person next to them, even in the same family. So you have to have the larger numbers to really be able to be confident in terms of, is it going to be able to help consistently, and who is it going to be able to help? And then you get FDA review after all of that, and if you're approved, you enter into what's called a phase 4, which is a continuous monitoring to make sure, because sometimes side effects take years to show up, and so you continuously monitor it. So the anatomy of a clinical trial, so that's the way the different phases, but what are the components of it? So first of all, what is your design? So what questions do you want to answer? In our case is, can we replace functionally a photoreceptor? But other people have different goals with their clinical trials, and that has to be clearly delineated. Size, how many participants do you want to have, or you're aiming to have? Sometimes you can have very small trials, or you can have bigger trials, depending on how common the condition is. Location, where are you going to do it? So that's very important. You want to have access to patients. You want to be able to make sure you can recruit. What are the inclusion or exclusion criteria? For example, an extreme example would be, you don't want to do this therapy in somebody who just had a heart attack a month ago, because if they pass away from a second heart attack, 4 minutes, very good. If they pass away from a second heart attack, was it your therapeutic? I mean, did that do it? I mean, is that a side effect of your therapeutic? Even though it seems like something in your eye wouldn't cause someone to have a heart attack, it's a reportable condition. So you try and hedge your bets in that regard. So there are inclusion and exclusion criteria,
And that really delineates who can be in the early phase trials. And that tends to change as you progress through different phases. Methods of assessment. How are we going to determine that A, it's safe, and B, it has some sort of an effect? And that's where we have a benefit in ophthalmology, because we have so many ways to test a person's visual function. And so those tests are baked into the trial at particular intervals, and that's carefully worked out prior to starting the trial. And then reporting. Who's going to oversee it? How do you have that arm's length review? So for instance, I can have nothing to do with any of the evaluation of the patients who are being treated. And that's important because I'm too close to it, right? So you bring in people who have no vested interest in the product or the process, and they're sitting there looking at the data and independently in an unbiased fashion determining, should we go to the next step? And so those are all really important safety factors that we have. And so that led ultimately to this phase 1/2A trial called Clerico. It's the first in the world for a primary photoreceptor replacement. And it's a partnership between us as cell manufacturers and Opsis and Fujifilm, and then our clinical partners, and it's been a great interaction that we've had, and we're really privileged to be able to work with really the two giants in iPS cell therapies, which is FCDI and then Blue Rock Therapeutics. But it was exclusively licensed by Blue Rock Therapeutics, so the trial is run by Blue Rock, and I consult for them, but it's, we manufacture the cells, but they run the trial. Okay. And how many trials this is a quick one, how many can I participate in? It's kind of important. One, right? And again, going back to most likely, and that's because we don't want to have confounding variables. So if you have a gene therapy, and then a year later you have a cell therapy, and then you get better, which one was it? Or was it a combination of the two? So at least early on, things being clean is very important. It's not just a thing that the FDA requires, it's important for the science of the whole thing. When and if it gets to approval, then you're open. I mean, once it's approved to be used, it doesn't matter if you had a previous gene therapy. It's between you and your doctor to determine whether you're eligible for it. And then are there risks? Absolutely there are risks. I kind of told you that already, so you have to consider these. There are surgical risks. Regardless of what we're putting under the retina, it's a surgery, so you have all the surgical risks. You have the medications that you're going to have to use. This is a transplant. There are immunosuppressive drugs you have to use to be able to keep the cells alive, at least for a short period of time. And so there are risks to those medications as well. Then there's the product. Those are two that don't even deal with what we're actually putting into the retina. But then the product itself, from a stem cell standpoint, because it can make every cell in the body, it's possible it could. So we have to do all the studies necessary to make sure we don't make beating heart under somebody's retina. But it should not be financial. So at this point, stage in the game, anybody that is charging you for a stem cell therapy, avoid, because they have a conflict of interest. Everybody that I know of that's doing legitimate stem cell therapeutics or testing them will pay you for your time. You are volunteering, you're putting yourself on the front lines for these therapeutics. And so, we pay your way. We make sure, if you have to travel, we're covering those costs. We're not going to pay you big money, because then that's a conflict of interest on your side, but you shouldn't be paying out of pocket. Okay, and then why does it take too long? And I'm just about done here. So why does it take so long? So hopefully by now you kind of realize this can't be done overnight. And our developmental timeline here from discovery, I start with 1997, where Jamie Thompson said, hey, we can make these cells, all the way through industry interest in 2016. That's all the work that we did in the laboratory and continue to do. Then the preclinical work, so doing all the work that we needed to do to get IND approval, so approval by the FDA to say, yes, you can do a clinical trial. And that's where we're at right now. So we're at the phase 1/2A in the lower left-hand part of the slide. And then we go on to a 2B, a 3, and so on and so forth. So it is a long time. And so why is it all necessary? And what I'm showing here is some switchback roads going down the Alps, right? So it's a lot easier to go straight down a mountain. It's a lot faster to go straight down a mountain, but there are risks to going straight down a mountain. And so what I'm showing here is my favorite analogy, which is, it's in Gloucester, and the Cooper's Hill cheese rolling race. I don't know who knows about this. I'm from Wisconsin. I'm interested in people moving cheese. So what they do is they take a roll of cheese, and usually very nice cheese. They roll it down the hill, and then hundreds, if not thousands of people just barrel down this hill, and whoever gets the cheese gets to keep it. That's your reward, right? So if you're really good, you get the cheese, yeah, you're great. But along the way, a lot of people get hurt. right? So, folks break their necks, they do all sorts of crazy things. And so the idea here is to be able to mitigate those risks and be able to make sure that at the end of the day, no one breaks their neck, and hopefully a lot of people get the cheese. And the eye is leading the way for the reasons I mentioned before. There's a lot of benefits to having two of an organ, having it be forward-facing, accessible, having the surgical expertise, and the outcome measures already in place, which de-risks the whole process. And so on some final thoughts, and I've already alluded to these throughout the talk, and that is, there's no technology that starts off right off the bat at its fully mature stage. So you think about computers and cars and airplanes. I mean, the Wright brothers started off by trying to push off of a sandhill and not kill themselves. And now we've got supersonic jets going across the ocean. So it's not realistic to think that we're going to, right out of the bat, give people back their driver's licenses. You have to crawl before you walk, before you run. And so I think there's managing expectations is an important part about any therapy, because you might generate a lot of enthusiasm earlier, but if you overpromise, you're going to underdeliver, and then people are going to be disappointed. What I have found is that in working with patients, that making meaningful improvements in folks' vision can be done in many different ways. It can be done just by giving somebody more light perception. It can be done by giving someone motion detection so they can cross the road a little bit better. And if we can make those meaningful improvements, then it will set the stage for more investment and more iteration in the technology, and then who knows where it will all lead and where the ceiling actually is. And along the way, be careful for the snake oil salesmen. Stem cells has a lot of snake oil, but it's not new. It's been around forever. If people can sell hope and hype and be able to fool you, they will, to make a buck. So make sure you engage FBC, engage your physicians, make sure you're getting good, honest information before embarking on any treatments. I want to acknowledge this is not my lab. I have no idea who these people are, but it's just generally thanking the hundreds of individuals that have been involved in this entire process. I can't name them all, but I'm just here able to talk about it. But it really has required dozens of people in my lab over the years, many, many dozens of people on the industry side, and I've just been really blessed and privileged to be able to have the journey. And thank you very much, and I'm happy to take any questions.
Thank you so much, Dr. Gamm. That was a wonderful, wonderful talk. And I looked it up for anybody wondering. The size of Corpus Christi is about the same size as Markham. So
thank you.
Yeah.
Can I switch that now?
Yeah, absolutely.
I have reason to switch from U.S. cities to
yeah.
Canadian cities.
So Markham, Vaughan, that's how many cells were in that container. Yep, so we've got time for a few questions. We have a couple of mic runners, Ushani and Kristen. So put your hand up, and I can see a gentleman over here. Kristen? Yeah, in the blue jacket. Hi.
Thanks very much for coming. My name is Milton Charlton. I was wondering, I'm sure, I guess you must have done this, you just didn't talk about it very much. Have you shown that the transplanted receptors make connections with the other cells, the bipolar cells, and that they are light sensitive and make an active light sensitive connection to the next cells?
Yeah, so what I mentioned earlier was we kind of did it in parts, right? So yes, they're light sensitive. They develop all the different structures. Not only are they light sensitive, but they're appropriate for the cell type that they are. If they're a red cone, they respond to red wavelengths of light, green, blue, rods. So yes, connections, that was also, so we published that in Cell Stem Cell. We published another paper that was talking about making the connections. And so the issue with the integration and restoring sight comes down to the fact that it's a transplant. So you have human cells, and if you put that into a rat or a mouse, just like if I had a human kidney, and I'm saying, well, I think kidney transplants might really help people. If I put that into a piece of a human into a rat, it's not going to work. They're developmentally and evolutionarily not compatible. So you have to kind of piecemeal the characteristics of the cell you're putting in, and then their behavior in the animal model. So in multiple animal models, so we looked at species from rat all the way to pig and non-human primates, and they behave the same in all of them. So one thing that was important for us to show is that they know what to do no matter what they're in. So if I have 16 different patients at different stages of their disease, if the cells do the same thing in a rat that they do in a pig, then having different humans shouldn't be an issue. And then the connection portion of it we were able to show as well too. So yes, all those things were done. Ultimately, getting a pig to read a newspaper by putting in a human cell isn't going to be done, right? And that's different from a gene therapy. So if you have an animal model and that animal model has a genetic defect that's similar to humans, and you replace that gene, then that's that animal's gene. So you should be able to see a strong functional readout. But for transplant purposes, cell transplant purposes, the bar is different for that. So all the pieces are there. Whether you put them all, whether they all come together into a human patient, we won't know until we do the human clinical trials. And I should say also that the clinical trial path is littered with really good animal studies that show all those things, but then fail in humans. So again, where we put our criteria, how we determine, and how the FDA determines what's worth a shot on goal is something that's evolved over the years.
Ushani?
Thank you for your presentation. It's very insightful. I'm interested to find out if there's any stem cells therapies research done on, say, for example, optic nerve damages. Is there anything on that area that's being developed or done right now?
Absolutely. And so, first of all, that's a much more complex thing. Okay, so the reason for that is one of the reasons why photoreceptors are neurons, and so they have to make neural connections. So it's a complex cell type. But the benefit you have is you can access that compartment directly. So you do a subretinal injection, and it only has to bump up to its next cell. So it has to extend, a wire, but very short distances. The optic nerve is composed of 1.2 million wires that are derived from the retinal ganglion cells, which are the cells that are on the surface of the retina. And so those have to go back to the brain. So they have to make connections that go down that optic nerve and find their connections in the lateral nucleus in the brain. So just from a biology standpoint, it's like spinal cord replacement. Okay, so just theoretically, the ability to replace those cells are much, much more difficult. That being said, we can make those cells. We can make retinal ganglion cells as well as we can make photoreceptors too. So, and there are groups. One of my first postdocs that was in my laboratory now makes RGC, retinal ganglion cells, and is interested in optic neuropathies. So he's spun off from my lab, and I kind of stay on my side of the room. He stays over there, but we talk a lot. More recently, though, there has been a call from the United States for whole eye transplant. Sounds crazy, right? I mentioned that a little bit early. But the key there is you have to cut an optic nerve, and you got to be able to put them together. So that brings to the forefront the idea of how do you make that connection again. And so what we're doing is not trying to replace the retinal ganglion cells in the donor eye, but make a relay neuron. So can we get the ganglion cells that are in the donor eye to make a synapse or a connection to a ganglion cell, and then have those ganglion cells, which have the ability to send axons a long distance, have them kind of act like, grab the baton and act that as the relay neuron for that. Still kind of a space shot, moon shot sort of thing, but there's a lot of work and money that's being put into those issues.
I had meant to say if anybody is watching online, and I know we do, if you want to put any questions into the Q&A, please do that. I think we have a question right now.
Hello? Yeah, so we have a
Morgan.
question. Hi.
Thanks for inviting me.
So good to see you. I know I didn't get to see you earlier. So we have a question from an online attendee. So they're asking, when an intraretinal injection is done, are the effects to be seen only in that particular area, or does it continue to work on the full retina area for photoreceptors or RPE?
Great point. And again, it's biology, it's not magic, in that area. So the cells, technically, so when you raise, it's like think of it as a blister. So you have the retina is kind of like a wallpaper on the back of the eye. There's actually a potential space between the neural retina and the RPE, so it's not like we have to peel things apart. When you have a retinal detachment, so if you are unfortunate enough to have a retinal detachment, it's always going to be between the photoreceptors and the RPE. It's where it happens all the time. It's an anatomic weak spot. We take advantage of that by being able to cleanly pull that off. So then you form a blister, and then the cells, they may be able to sneak their way a little bit past the borders of that blister. For the most part, they can't do that. Same thing with gene therapy, by the way. Only recently have there been some gene therapies that have shown some ability to laterally spread beyond what we call a bleb, or the blister that's made.
Okay.
And that's a limitation, right? So you can only treat that area. So can you do multiple treatments? Yes, you can do multiple treatments, but not initially, right? So Luxturna did that. They did gene therapy. They treated one small area in the retina. It was safe. People saw a little bit better. Then they allowed them to go back in and do another bleb.
Okay. We've got a question over here.
Hi, my name is Mohammed. I'm a RP patient, advanced stage. I have only the light prospection, like a light blinking in front of my eyes. Actually, I tried 10 years ago looking for the trials, approximate trials. I enrolled in the NIH, National Institute of Health. I enrolled with Dr. Henry in California. So whatever. So NIH tested my gene in University of Utah lab. They find it's not genetic. Then when I'm in Canada, my ophthalmologist tested. And they, in California, one of the gene lab, they also find it's not genetic. It's some unknown reason. So my question, because I see Blue Rock, like other trials, they're doing E64, the genetic one. But there is any, like a trial 4 or trial 3, is for unknown reason, unknown gene, anything going on for the unknown reason. So if it's working for the unknown reason, this trial 4, so basically I'm looking to enroll myself as a patient. And I'm trying my level best. I also talk with the Blue Rock in Toronto Center. There is an office in Blue Rock. And
let me just jump in there too. So I think I can answer your question, and then, so first of all, it doesn't mean it's not genetic. Because they didn't find your gene does not mean it's not a genetic disease. Only about 2/3, maybe 3/4 with some of the chips now will be able to detect the genetic cause of a person's vision loss. So just because you can't find it doesn't mean it isn't genetic. What we're finding is that the parts that can go wrong in the coding of proteins don't have to be in the actual protein itself or the protein coding part. It can be in areas in the vast wastelands of our DNA that control how they're expressed. And we're just now getting a better handle on that. So, but it's still, you end up in the same spot. You still end up without a genetic diagnosis. Early phase trials, we want a genetic diagnosis. We want to know that you didn't lose vision because of something that wasn't genetic. Maybe you lost it because you had an infection or you had an inflammation or something like that. Usually we can tell that by your clinical progress. So if you have a family history of vision loss, or depending upon when you started, what your symptoms were, how you progressed over time, we can be pretty clear clinically whether or not it's an inherited disorder or not. And so if that's the case, if you have a clinical picture of an inherited retinal degenerative disease, but you don't have a genetic diagnosis, keep trying, because the technology gets better and better every year. So keep talking to your eye care professionals.
That's why I'm trying to looking that.
Yep. And then lastly, the answer to your question is yes. So once you get to like maybe a phase 3, that sort of thing, then most likely they'll open it up to folks that have clinical diagnoses and not just genetic diagnoses. And so yes, you won't be forgotten. And if it's approved, then it's on label, and it's a discussion between you and your retina surgeon.
Yep. Okay. We try our level best, because my ophthalmologist also looking here, there. Myself, I'm looking here, there.
Yeah. So hopefully I answered your question. Yeah. That you're doing all the right things. Just keep that dialogue going with your retina specialist, and make sure you're talking to somebody who's an ocular genetic specialist.